section 3, which deals with control of the cell cycle. So we'll start with a little review of the cell cycle. We know that the cell cycle is what one cell goes through during the course of its lifetime. And what we've been talking about for a cell cycle are those cells that do divide. So the end result of the cell cycle is two cells that are genetically identical to the cell that you started with. The first part of that cell cycle we said was interphase. And interphase was divided into G1, normal growth, S, making an entire copy of the DNA in the form of DNA, something that only happens during S, G2, last minute growth, and from there we went to mitosis. In mitosis, we divided into prophase, metaphase, anaphase, and telophase. We called it P, M, A, T, PMAT. And that was followed by, a little exaggerated here, but cytokinesis. We said right along that during interphase, the DNA is in the form of chromatin. I'm going to write T-I-N this time, so we're all clear. And in fact, when we leave telophase, we're back in the form of chromatin. Let's change colors if we can. Back with it. In mitosis, our DNA is in the form of chromosomes. And those chromosomes in prophase and metaphase were two sisters joined, and in anaphase and telophase they were one sister chromatid. What we learned from this last section is that all along this pathway there are places where there are stop signs. And pardon me, I cannot draw stop signs. Hexagons are not my forte. Those stop signs we'll call checkpoints. And those checkpoints are places where the cell stops what it's doing and decides whether it's safe to go forward or not. In the cell all the time, there are I went to a new page here. In the cell all the time, there are cyclin-dependent kinases, and if the cell is ready to move through that stop sign, they make cyclins. If they are not ready to do that, they will not make the cyclins. So let's get a new page up here. Don't save. Cyclins produced when ready. Cyclin-dependent kinases are always present, usually off. They are only turned on when cyclins are produced. So when cyclins are produced, the cyclin-dependent kinases are activated and the cell moves through the cell cycle, through the next part of the cell cycle. Let's see. Why is that? Well, let's take a look at a stem cell, a skin stem cell. That stem cell goes through the cell cycle. When it's all done, you will have two cells. You will have a skin cell and a skin stem cell, which is important because we've got this skin cell, is going to replace a lost cell. The skin stem cell replaces the stem cell that gave rise to the skin cell. So in the end, we don't have any new cells. We lost one skin cell, and we started with one stem cell. So we gained one skin cell. And if we have to do it again, we can do it again and continually replace those lost skin cells that are falling off all the time. And we don't have any growth. There's no change in the total number of cells on that piece of skin. If the cell cycle doesn't work correctly, and suppose this skin cell, let's put a new one up here, that skin cell, that stem cell divides, and we get a mutant skin cell. That mutant skin cell may now have the ability to divide. So, if it becomes cancerous, that mutant stem cell, mutant skin cell, will be two skin cells. Those two skin cells both have the ability to divide. Those four cells also have the ability to divide, so that what you end up with is instead of one cell replacing one cell, we now here have 1, 2, 3, 4, 5, 6, 7 cells replacing one cell. And those 7 cells are not normal. They have the ability to divide whenever they want to, and that is how we might develop through numerous mutations. Remember, it takes more than one. They'll have to lose control of the whole cell cycle. A skin cancer. And that's the real danger. Those skin cells are going to push out of the way other cells. Eventually, those mutant skin cells won't stay where they are. They will go somewhere else when they become metastatic and go into other tissues where there really isn't any room for them. So we really want to keep the cell cycle under a very tight control, and we do that using genes. And if those genes become mutated, cytokines and cyclin-dependent kinases, we can end up with real trouble. Now, we also talked about in this section 2 other kinds of cells. One of them we just touched on, and that is our old friend, the stem cell. Stem cells have the ability to, 1, reproduce themselves, so we never run out of stem cells, and 2, they have the ability to become something else. In the case we just talked about, a skin cell. In other cases, we have them in our intestines, we have them in our blood. They can replace all of our blood cells. And there are many other examples of these stem cells in our bodies. And these stem cells are called adult stem cells, not because they're found in adults, but because they are limited in possibilities. The other type of stem cell that we have to be aware of is the embryonic stem cell. And these are unlimited in their possibilities. They can become any other cell, which means there's a lot of research going on to heal disease using embryonic stem cells. But the easiest place to get them from is an early embryo, and most of those come from fertility clinics, because parents want to or have to, in order to have children, have in vitro fertilization occur. So a sperm and an egg are mixed in a test tube. Actually, thousands of sperm and thousands of eggs are mixed in a test tube, and those that fertilize are frozen away and later on can be used to continue a pregnancy. So these embryonic stem cells, while they might have the ability to heal disease, they are each a potential person, and that makes the use of these quite controversial. The other topic we touched on in 9.3 is apoptosis. This is a genetically programmed cell death. There are times when it can be used as a way to prevent cancer, and it's certainly a very important part of development in multicellular organisms to get rid of cells that are not part of the final adult organism. All right, stay tuned tomorrow for a overview of the whole chapter, after which I will post a review quiz on Classroom that I would like you all to take.